As part of class, we'll cross two worm lineages, or populations, and then mate their F1s to produce an F2 generation. Then, if there are incompatibilities between the parental lineages, we'll expect some genotypes to be missing in the F2 generation. For example, if there is a recessive interaction between the red and the blue alleles of these genes, then we would not produce genotypes in the upper left corner of the Punnett square. So, we need to collect F2 offspring and genotype them. Because you're not in the lab, I will perform these crosses and genotype the F2s. You do have an active role in this process, because there are decisions about the experiment that you should make. We have a hurdle in this type of research project, because if an incompatibility exists, we don't know which pair of the 20,000 genes across all 6 chromosomes are the incompatible ones. In most research projects of this type, the genotype of each of hundreds of F2 individuals would be collected at each part of each chromosome, and then we would look for missing genotypes, considering every possible pair of genes. That's 20,000 times 20,000, or 400 million comparisons. Instead, because of time and cost limitations, we need a more limited approach. We need to pick perhaps one gene per chromosome to genotype a small number of F2 offspring with. Also, there are many different P0 populations that we can work with, so we need to select perhaps 1 or 2 pairs of populations to cross. We will provide information about the choices of worm populations and which loci we can genotype, so that you can make those decisions as we get started with this research project.